Morning, everyone, to the second day of our PBBM Global Workshop. And we have a full program again today. We're going to start out with a survey that Javier had put together and most of you filled out. So we will take a look at what the barometer said about in the room about the PBBM model applications and experiences. So Javier, I'll turn it over to you before we go on with the other program.
Thank you, Tycho. So I'm not going to take too much of your time. It's just to reinforce, the need to have as many respondents as possible. So if you have filled the survey, this is perfect. Thank you very much. You're going to see just a snapshot of the current results, but it's not over yet. There's still 2 days to come. So please take 10 minutes of your time to fill in these questions. It's really important because that will be used as part of the publications which will come out of this exercise. So just to give you a brief snapshot of where we are now, we've got 76 respondents, which is good. Most of them, half and half, the top respondents come from the pharma industry and the regulatory authorities. We've got, and the rest is here, you can see it. So 1/3 of the attendance has had experience in either submitting or reviewing a PBBM. The 2/3 didn't. The other 2/3 didn't. And the main reasons why they didn't is that they didn't have any data to share or to submit. And there's also another. The feedback of the survey is in French, so that the last bottom means other. If you didn't know French. Then the question 4 was about the type of drugs that was used for the PBBM, and it's mostly oral IR product. The topics that were covered in the PBBM, so you can see the majority comes from justification of the drug product dissolution spec. So with lots of examples there. Have you encountered or recommended rejection of a submitted model? So the current situation is, 2/3 of the submitted models are being rejected. And the reasons why they are rejected is that they do not contain non-BE clinical data. And the second top response is that it was not fitting the clinical data adequately. So it failed validation. And I think it's interesting to have these questions here, or these preliminary answers, because today we're going to talk about model validation. So it's really to the point. Then there was two series of questions on the risk ranking. So the risk ranking in terms of regulatory decision, risk associated to the patient for question 8, and the risk associated to the or the chances of regulatory acceptance onto the right. So again, I won't go into any of the details here. That's an interesting one. So assuming a full PBPK model was developed and the PBBM was validated on rich PK data during phase 1, including SAD, MAD, 1 food effect study, 1 variant study, what would be the recommended average absolute prediction error for model acceptance? And here the majority is proposing less than 20%. Finally, could you have different criteria for AUC and Cmax? Yes, with justification. And should the clinical variability be considered to set acceptance criteria? Yes. And is there any value for industry to develop a PBBM template? Yes, majority. So that's it. Thank you very much, and please fill in the survey.
All right. Switch the slides. Thank you. And yesterday, the day was mainly about inputs into PBBM models. You heard about precipitation, you heard about permeability, you heard about our dissolution profiles, and how to model dissolution data, etc. And today is more along the lines of setting up base models. So there will be, as yesterday, case studies presented. But before we go there, I wanted to talk again about this particular figure here, because we took quite a while to get the alignment that the PBBM is a subset of PBBK. So if you call the whole workshop here PBBK, it's all right. It's still mathematically or whatever correct, but it's just not very descriptive. That's like saying a car when you drive a Ford, a Mustang, or any other model, and it is still correct. It is a car, but it is not descriptive. So if you think about you wanted to be specific in what type of modeling you do, I think you're better off to call it PBBM if you're supporting drug product quality. Today, the case studies will be again presented by regulators, and I will have one case study as well from industry, talking about setting up the safe space, and also some of the challenges that we have when we talk about impact. I did want to make the comment about impact. Yesterday, regulators did a very fine job with looking at impact, and the majority were of low impact, meaning from a regulatory point of view, it was not big impact. But within the companies, the PBBM can have huge impact because you're selecting formulation, you're saving money because you're choosing the right formulation to go into manufacturing. So I think there's probably some aspect where there's maybe a different perspective about what constitutes high impact. And Xavier talked about the dissolution specification setting just in the survey, right? If you use totality of evidence together with PBBM to set the specifications, then that can have, at least for the company, a significant impact in terms of cost reduction, which I like to think ultimately will also benefit patients. So going to the next slide, you will see the agenda for the day. As before, there is a roundtable with regulators. The way the roundtable will run is we have pre-prepared questions. Claire and I will be moderating. If there's time at the end, audience participation is highly encouraged. We want this to be relevant and lively. And the session this morning starts out, I'll give a case study, and then we have the other case studies presented as well. I do want to remind everyone that part of this workshop, we are going to have 4 publications at least. So there will be 1 for each day. It's very important, please, that the scribes and the moderators ensure that the points we're discussing are captured. The purpose is not to have 100% agreement, and the purpose is also not to have recommendations. We can have something that's called points to consider when you're doing a modeling or talking about model applications. So it's important that we capture what was discussed here for people who could not attend the conference, or for people who just want to learn what is being discussed. We would like to have the draft manuscripts for each day. I would like to say next 4 to 6 weeks. I know Jim had the dissolution workshop earlier this year, and he said it's doable. So I'm hoping that we can be as good or better. So I just wanted to remind everybody, scribes, please be meticulous and capture the inputs, what's discussed. I think with this, we can probably go to the next slide. And again, the breakout sessions, everybody is encouraged to participate. Do not be intimidated. It is a friendly audience. And please make sure that you contribute to the topic of your passion. Again, as we had last year, yesterday, your pre-assigned room is on your badge, should be on your badge. And then we will have later on in the day, we have the summary sections as we had yesterday as well, will be moderated by me and Kim. So with this, I think I'm going to start out. I think Kimberly has a couple words to say.
Good morning, everyone. Welcome back to day 2. I think yesterday we had a lot of lively discussions. I think Tycho covered pretty much everything in a summary, but the only other thing I would say is this is a unique opportunity for us to engage and talk with each other. So take opportunities. If you had remaining questions from the breakout session yesterday, speak up to the hot topic leads and encourage that dialogue. So without further ado, we'll get started for today's presentations. I'll be introducing Tycho, which really doesn't need an introduction, but Dr. Tycho Hembeck is a senior principal scientist at Merck in the Biologic Department and Biopharmaceutics Group, which is part of the Sterile and Specialty Project Groups. There he serves as the biopharmaceutics and PBBM, PBPK expert in oral and long-acting injectable drug development, which includes establishing the bioequivalent safe space of new drug candidates. Prior to that, Tycho was a director in DMPK at Novartis, where he led a global PBPK group and served as the PBPK and biopharmaceutics expert and implemented PBPK, PBBM for oncology drugs. Tycho has served as co-chair on working groups for the PBPK modeling and PBPK renal and hepatic impairment working group and the PBBM working group for innovating in quality and pharmaceutical development consortium. He is currently serving as Merck's representative on the PBBM Innovation and Quality Consortium Working Group. Dr. Hembeck has been a speaker at 50 national and international conferences. He has authored and co-authored over 65 peer-reviewed publications in anatomy, PBPK, and formulation science, and was recognized as an AAPS Fellow in 2021. So welcome.
Yes, thank you. Thank you, Kimberly. I guess I have to shorten my introduction a little bit for the future use, but I will be concise here in the case study presentation. I just wanted to talk about a poster, and you can't read this, but you'll get the slides, and I have blow-ups of what you're going to look at. So molnupiravir is a Merck drug that was used for COVID-19 treatment and authorized under emergency use authorization. And we had to set up a dissolution safe space because manufacturing occurred in various sites because it's a high volume product. So what is molnupiravir, right? Molnupiravir shown on the structure here, you can see it's an ester, and it's actually an isopropyl ester, which, as you will see in the future slides, gets hydrolyzed, and this is the prodrug. The purpose that we had was to characterize molnupiravir absorption in systemic pharmacokinetics, and then using multiple clinical study data to look at the pharmacokinetics and to describe the mechanism of absorption, but also look at various capsule formulations and solution data. So the data I'm going to be showing was partially based on the Painter et al. Study in 2021, which is the Ridgeback company, which we collaborated with. Now, right up front, the solubility of the product is very high. You can see it's 43 to 89 milligram per ml over the intestinal range. And even at the dose of 800 milligram, the dose number is less than 1. Therefore, it's high solubility. So that's a slam dunk assessment. Where we have challenges is with the permeability. And I was glad to see the permeability discussion yesterday. We assessed the intestinal permeability with the rat intestinal perfusion model, which had originally been part of an FDA guidance. And in that rat intestinal perfusion model, molnupiravir had a higher permeability than metoprolol. Metoprolol, as you know, is a reference compound for high absorption with a known fraction absorb higher than 95%. And in the rat intestinal perfusion model, molnupiravir has demonstrated very high permeability. The reason that's important is because we're using this very high permeability in the as an input into our PBBM model, which is GastroPlus. Please know that the fraction absorbed was also high in radiolabel study, not in human, but we don't have a human study, but in rats. So we believe the compound has high intestinal permeability. And this is a little bit of a busy slide, so I'll explain it a little bit step by step. So in the lower right corner, you can see the prodrug in red. We call it molnupiravir mauve. And then you have the metabolite, which is called N-hydroxycytidine. It's a nucleoside. And you can see that we're showing it here in blue. It's called NHC. Now, in the clinic, in all clinical studies, we cannot detect molnupiravir in the systemic circulation. So the data which you're seeing is the open squares, is the clinically observed data, and it is the metabolite NHC. There is, again, across all studies, no molnupiravir that can be found in the systemic circulation. So what does that mean? It means that molnupiravir is a very effective prodrug which very rapidly generates NHC. And if you look on the top, the top from left to right is from 200, 400, and 600 milligrams. So this is powder in bottle solution data. And the Cmax, Tmax is early. It's between 0.75 and 1.5 hours. So it's, again, a testament that the conversion is very rapid. Now, you will see that there's additional lines added. So if you look, for example, at the 400 milligram powder in bottle dose, you can see that we are calculating 394 milligram reach systemic circulation. That's basically on the diagnostic plots using the mass balance that's available from GastroPlus. And we can also see that there's a significant amount, I think it's 292 milligram of NHC formed. Mathematically, you can only get 314 because it's a one-to-one conversion. So the conversion is also very efficient. We're calculating 292 out of 314 milligrams. So you can calculate your fraction absorbed from that alone. So that's, again, very consistent with high permeability. And then you can see that we have captured on the top, again, the solution data, and at the bottom, we're just showing some representative data from capsule data from the multiple ascending dose study that were done on day 1, and again, capsule data. In general, the compound, and that's now being published, has been published, has linear pharmacokinetics in the developed formulations up to 1,200 milligrams. And I should point out, we have since also generated food effect data. There is no food effect for molnupiravir. And here is a little bit the decision tree about what we did. I know this is a little bit busy slide, but it's as always model setup, model validation, and then model application. So the application was to be able to look at dissolution profiles from different manufacturing sites and determining whether or not those would be leading to bioequivalence, because some of the batches had faster dissolution profiles than others. And so we were looking at Cmax, as you will see, as one of the criterion. So, but if you look in the upper right corner, you can see that we are using the high permeability as input. And then we talk about clearance. Okay, clearance and volumes, the systemic PK parameters. I know yesterday, most of the studies had the Mercedes-Benz of data, including either microdosing or human IV data, or even multiple IV data. Here we don't have that. Okay, we are not going to have IV data for molnupiravir. But what we do have is very consistent PK predictions, because the nucleoside is readily detectable. And if you scale from rat to human and you scale from dog to human, the human clearance is identical. And so on top of that, we had the population pharmacokinetic data. So we use the bottom-up approach from animal data and combine that with pop PK data. And that's how we come up with the human clearance parameters. Clearance is moderate, and bioavailability estimated is also moderate, consistent with preclinical data. So this is probably one of the perceived weak points, but I don't think it is a weak point necessarily, because allometry can be used in certain cases. For example, if you look at volume distribution in non-nucleosides, you look at all the nucleosides that have ever been in drug development, the volume distribution is very consistent. It's about between 1 and 1.5 liter per kilogram. So you can even use a QSAR model for that. So then if you go down and look over again, we have the solution data, various studies, and then we have also the capsule data that's included. I didn't include the food effect data, but it's also part of the validation. And then we have at the bottom, we have two independent studies, completely independent studies. They were done in patients. And one of them actually had a sparse PK. So we didn't have a full AUC, but we have a Cmax. Now you see the criteria we used was less than 25%. You just saw the survey. Most people prefer 20%. We listed 25% as our criterion, but most data were actually less than 20%. They were typically 15%. And I know the newer data that we have is even tighter because we have an improved formulation. So then you can see at the bottom that we have the safe space as an application. And this is done with the Z factor. It's using dissolution requirements methods from the FDA. And you can see that because we had solution data and we had, again, the capsule data, what you're looking at, the open squares is the reference capsule, and the capsules that we ended up making from at Merck at different sites, they were either equal or faster. So the concern was, are we seeing a significant increase in Cmax? But when we did the modeling, we found no significant impact. So we were basically concluding that the pharmacokinetics are not altered and independent. And this is my last slide. I can tell you we have since expanded the safe space, and we have used a new formulation variant, which is below the dashed line, because we use a PBBM to inform the clinical studies. And we use a formulation variant below the dashed line, and that formulation variant was still bioequivalent. So to all of those who say that we must have non-bioequivalence data, I challenge you, because sometimes it is difficult to even make a non-bioequivalence batch. And so we have data now that is showing bioequivalence below the dashed line, which we had originally calculated. And then, of course, from a regulatory point of view, we can basically hopefully argue that because we have clinical data, the knowledge base is there. It goes beyond the green line. I wasn't allowed to share the data yet, but we have it. And so that's how we use a PBBM in drug development. I know I'm probably up for time, but in the breakout sessions, I'm going to be talking about QAW0039, fevipiprant. If somebody is trying to learn how to use GastroPlus or how to do the analysis, I recommend you read this paper. It has 70 pages in the supplement. You can find out in detail how you do parameter sensitivity analysis for the Weibull function, the slope factor, the percent release, the POP-PK. It's really described in minute detail, and it's a very powerful study. This is a study that probably regulators like to see. It has microdosing data, it has BE data, it has non-BE data, and it has 2 different dose strengths. So that's the full package. And I'm gonna stop now, and then we can see with the next program. Thank you. I'm going to introduce the next speaker. It's now my pleasure to introduce Mary Malamatari. She is representing the MHRA, and there she is a clinical pharmacology assessor in the Department of Innovative Medicines. Dr. Malamatari has received her undergraduate degree from Aristotle University from Tisaso Cooley, and has carried out her PhD in the Center of Doctoral Training for Targeted Therapies and Formulation Sciences at the University College of London. And she comes here with significant experience, so I'm glad that she can present one of the case studies. Thank you.
Thank you very much, and thank you for inviting me in this conference. So we will see case study 5 by Janssen. Here for the next one. Okay. Here is the standard disclaimer. Some introductory slides that PBBM modeling emphasizes the integration of the physicochemical properties of the drug substance and formulation characteristics with the system physiological properties to predict the absorption of a drug product. And here is the overall workflow for a PBBM, where we start with the model objectives that they should be clearly defined. Then the next stage is the stage of model development, where focus should be placed on in vivo dissolution and absorption. And we have a mechanistic absorption modeling with a simplified disposition model. The next stage is the stage of model validation, also known as predicting the known, that the validation can be internal, using data sets that have been already used in model development, or external, using new data set. And the last step is the step of model application, known as well as predicting the known. For this case study, the drug substance was a weak base. It is classified as a BCS class 2 compound, meaning that it exhibits low solubility and high permeability. The drug product is an immediate release solid oral dosage form, and as per the product information, it should be taken with food. The problem, the issue in this case was that 2 batches, let's call them batch X, when tested on stability, they showed out of specification results for the QC dissolution testing method. All other stability tests conform to specification at shelf life, and no root cause could be identified for the out of spec results. So the strategy used was to develop a mechanistic absorption model, validate the model using a relative bioavailability study that we will see in more detail in a bit. And then the model application was to assess the clinical impact of failing dissolution specification for batch X on stability. So during model development, the 4 inputs were can be categorized as compound-specific inputs like solubility and permeability, formulation-specific parameters such as dissolution, physiology parameters, and the gastro plus the human physiological fasted or the human physiological fed model were used. And lastly, PK parameters such as the clearance or the volume of the compartments. And all these inputs were fed into the model, and the model output was the PK profiles, and namely the AUC and the Cmax. Regarding the compound-specific parameters, solubility was measured in aqueous and biorelevant media. And here in the graph, we see the solubility pH profile of drug fitted with the Henderson-Hasselbalch equation by Castroplas. In vitro permeability was not predictive of the in vivo situation due to extensive cellular binding. So instead, the effective permeability was predicted using the ADBET predictor tool and was used in the mechanistic absorption model. A point here to make is that normally we prefer measured parameters. However, predicted parameters can also be accepted as long as they are justified and they are accompanied by a parameter sensitivity analysis. Now, regarding the dissolution, the applicant has used the physiology-based dissolution testing, the PBDT, that it is a 2-phase dissolution where initially the formulation is placed in phosphate buffer 300 ml pH 4.9 and dissolution take place for 60 minutes. And then the formulation is placed in the second phase that it's FESIV 900 ml for 3 hours. And these setups simulate the physiological GI conditions in the fed state. However, we noted that there was no clear link between the physiology-based dissolution testing and the QC method, that the stability batches were out of spec. Z factor was used as the mechanistic way for integrating the solution to the model. The Z factor that has values of volume, has units of volume per mass per time, is a composite parameter function of the drug diffusion coefficient, true crystal density, thickness of the unstirred water layer, and the inner particle radius. We would like to see more information on how the Z factor was fitted to the data. For example, were both the rise and plateau of the solution captured in the calculation of Z factor. Also, we would like to see a discussion on the limitations of Z factor. And yesterday in breakout session C, we discussed and that sometimes Z factor may not be used, it may be problematic to be used in micelle containing media. And here we have the FESIV. So we would like a discussion on the limitations of Z factor. Also a parameter sensitivity analysis on the effect of Z factor on exposure was not provided. Justification should be provided on the method selected for the solution data input. And yesterday, again in breakout session C, there was a decision tree on which method will be used, PPSD or Z factor. And this decision tree could potentially help to justify the method selected. And lastly, because the physiology-based dissolution testing that I described before was mimicking the fed state, clarification and not the fasting state. Clarification should be provided on how the Z factor in representative of the fasting state was obtained. However, from now on, I will mainly focus on the fed state. The Z factor can be used as an input option in several commercial softwares. And based on the literature, it has been used as a dissolution input method for pH-dependent drug-drug interactions, for justification of biowaivers, and also establishment of dissolution safe space. For the pharmacokinetics model, no IV data were available, and a 3-compartmental model was derived from low-dose oral pharmacokinetic data obtained after dosing a solution in healthy volunteers under fed conditions. Model validation was performed using a relative bioavailability study where healthy volunteers received a single dose of 100 milligrams under fasted and fed conditions. And as I said before, we will focus on fed conditions because a significant positive food effect was observed. And here we see at the table with the formulations used. There was the reference formulation containing disintegrin 1, and there were another 2 formulations containing another disintegrant. The one had drug substance with fine particle size, and the other one a drug substance with coarse particle size. And we see the results of the clinical study, of the relative bioavailability study, and both formulations are not bioequivalent to the reference formulation, apart from the AUC 0 to 72 hours that was found to be equivalent between the formulation with the fine drug substance and the reference formulation. So the applicant performed the physiology-based dissolution testing on the 3 batches, and we see that there is no dissolution in the first phase of the phosphate buffer. And then when the formulations are placed in the second phase of FESIV, we have a rapid dissolution where the dissolution rate of the reference formulation is high, followed by the formulation with the fine drug substance and the formulation with the coarse drug substance. And based on the physiology-based dissolution profiles, the Z factor was generated for each formulation, and then it was fed into the model, and we have the mean simulations for Cmax and AUC, underfed and fast state. And once the variability seen in the clinical study was incorporated to the model, the population simulations were generated. Regarding the variability, we would like to see additional information on how variability was incorporated in the model, and it should be discussed how the variability incorporated in the model affects the upper and lower levels of the geometric mean ratio in the virtual crossover trials that we will see in the next slide. So here are the results of 10 virtual crossover trials, and the results replicate the results of the clinical relative bioavailability study, meaning that the two formulations were not bioequivalent to the reference formulation, apart from the AUC 0.72 hours for formulation with a fine particle size and the reference formulation. And then going to model application, as we said, the objective was to assess the impact on exposure of not meeting the QC dissolution method at stability, at shelf-life. And the comparison was made with a reference batch on stability that was in specification and which was the commercial validation batch. Again, here now during model application, the physiology-based dissolution testing was performed, and we see again no dissolution in the first phase and a rapid and similar dissolution for both batches in the FESIV. Again, based on these profiles, the Z factor was generated for its formulation, and then the mean simulations were generated, and the variability seen in the clinical study was incorporated in the model, and the population simulations were generated. And now, based on 10 other virtual crossover trials, we see that the two batches are bioequivalent. The batch X that was out of spec for dissolution in stability and the commercial validation batch were bioequivalent both for Cmax and AUC. Moving to conclusions, with a mechanistic absorption model was developed using the Z factor as input method. The model was able to differentiate between tablets that were shown to be bioequivalent and non-bioequivalent in healthy volunteers in the relative bioavailability study. Virtual bioequivalent trials were performed, comparing the out-of-spec batch with a reference batch, which were predicted to be bioequivalent for both Cmax and AUC under fed conditions. So we can conclude that no impact on drug exposure is expected for batch X, for which the out-of-spec dissolution results were obtained using the QC method during stability. However, as mentioned before, no link between the QC method and the physiology-based dissolution testing was established, and no safe space was demarcated. Lastly, some take-home messages is that the model objective should be clearly stated. Predictive parameters, as we said, can be used as if justified and followed ideally by a parameter sensitivity analysis. Use of biorelevant dissolution method is highly recommended. Justification should be provided on the methods for dissolution data input, for example, why the Z factor is used instead of the Johnson method or the PPSD. The limitations, we want a critical discussion on the limitations of the method selected for dissolution data input. Also, a discussion should be provided on how variability is incorporated in the model and how it may affect the results of the virtual bioequivalent studies. Wherever possible, a safe space should be demarcated. And lastly, use of the checklist for PBBM regulatory submissions is highly recommended. Here is the reference, some of the reference used, and I would like to thank you And also, I would like to thank my colleague Sue Cole that we did together the work, but she was not able to come to this conference. Thank you.
Thank you. It's very nice presentation. Thank you. So my question is that after you have demonstrated that actually the OOS batches actually are also bioequivalent, bioequivalent. And then what's your next step regarding resetting the dissolution method?
Maybe use microphone.
Yes, I don't think I'm the right person to reply to this. I think something maybe that I didn't highlight is that it is interesting how the model objective was set in this case. If we consider the model objective being batch X, the impact of not meeting the QC dissolution method on exposure, then we accepted this. If it is that whichever batch doesn't meet QC dissolution method, we can consider the impact on exposure to be low, then this we wouldn't accept it, if I can make it clear. Now, what was your other question?
QC dissolution method. You just use the current. Sorry, so your approach is that you are going to use your QC method, the old one, but whenever there's OOS, you're using your modeling result to justify whether it's BE or non-BE. Is that the approach you're taking, or you're modifying your QC dissolution method to make that reflecting the BE situation better?
Yes, I know this is an interesting question. I think maybe from Janssen, they may want to respond. I think, yes, the physiology-based dissolution testing was much more discriminative, but we understand that the QC method on the same time should be easy to be used and simple.
Yeah, I'll do my best to answer the question. So indeed, this particular product was actually put on the market quite some years ago. It was actually before the utility of PBBM came in. And actually like Mary explained in the case study, it was on ICH stability, so long-term stability, that we saw this out of spec. But obviously, because of the commitments, we have to explain why we're out of spec. So that's why we use the model. So to answer the question, we still carried on using the QC method. And we did go into discussion with the various regulators to also modify the Q point and also the time point. So we were able to use a combination of the model and also product knowledge to be able to reset the spec based on what we saw. But the whole point here was to try and demonstrate that the QC method that had been developed, say, 5-8 years ago, was actually over-discriminating. And that's what we wanted to suggest. However, we also have to be transparent on the data we have and explain what we saw. So I hope that answers your question.
So again, long-term goal is that still you're going to modify your spec and or the dissolution method, right?
The QC method will stay for this point in time because the QC method is built per drug product, and the drug product the formulation or something like that hasn't been changed. If for some reason we were to build a new formulation, why? Then, of course, we would consider to rebuild the QC dissolution method. The current QC will stay. We will monitor it. It's only that one batch that has ever gone out of spec. Everything else is sitting super nice, super tight. However, we had to explain why and what we thought was happening.
So this is a very rare case, basically, the OS. Okay, gotcha.
Sorry, guys, I know there was another question, but we have to hold them to be on time. Thank you.
Thank you. Yes, we should still have time during our panel discussion for additional questions. So now I'll introduce the speaker for the next case. Dr. Luisa Burgos is a pharmacist with a specialization degree in clinical research from Harvard T.H. Chan School of Public Health. Working as a regulatory affairs since her graduation, she's been at the Brazilian Health Regulatory Agency, Anvisa, since 2014. In the General Board of Medicine, she's worked as a reviewer of clinical and preclinical studies for new molecular entities, and presently acts as a reviewer of bioequivalence and DDI studies and as an inspector of CROs. She has participated in global regulatory harmonization initiatives representing ANVISA in the International Council of Harmonization as an expert in M12, drug interaction studies, and M4E benefit risk information working groups, and in the Center of Innovation and Regulatory Sciences initiatives on benefit risk assessment and reliance pathways. Currently, she leads the initiatives in ANVISA regarding biopharmaceutical applications of physiologically based pharmacokinetic models. Welcome.
Good morning. I will present on PBBM case 4, EMD compound A from M-CERSI, Germany. Starting from the disclaimer, this presentation reflects my own views and should not be construed to represent ANVISA's views or policies. This case study were presented as a mock case study. It was not a real regulatory submission. The analysis were based on the report version 3 submitted in June 2022, and also as appropriate, some new data, some data discussed with the company representative on November 2022 were also included. I will use the yellow boxes to highlight some questions from the PBBM template that guided the analysis. EMD compound A is a hydrochloride salt compound. It is a BCS4 used for the treatment of certain cancers. Approved posology is 500 mg once daily under fed conditions. And to increase exposure of the compound, it is manufactured with the API in a micronized form. So the regulatory question proposed in this case is, does the agency agree that the acceptance criteria for the drug substance particle size distribution as the 10, the 50, and the 90 can be justified on the basis of the PBBM approach, or does the PBBM only qualify for supportive data? Current specifications for the product is approved based on results of the clinical studies, drug substance batch release data, and the manufacturability of the drug product. On the blue table, the proposed specifications based on PBBM are highlighted. They are based on PK results of TF2 formulation, which is not the market formulation, but is a formulation that is bioequivalent to the TF3 formulation, which is the market formulation. Manufacturing process of both formulations are dry granulation, followed by tablets and coating. But further details on the differences in TF2 and TF3 formulations were missing in the report and would be necessary for regulatory submission. Here, I present the workflow presented in the report of the modeling strategy. Companies starting to build the PBBM model bottom up, and they saw an issue that I will further describe with the Tmax. So then they incorporated lysosomal trapping on the bottom up model and called this as optimized PBBM. This model went through the validation, including 5 validation data sets, and were considered validated and then were applied. So data used for model building were data from the IV formulation and also oral solution formulation. Solid formulations as tablets and capsules were not used for model building, but were used for validation and parameter sensitivity analysis. On the presentation, I don't highlight this data, but on the report, there is more details about the bets of APIs and the bets of the drug product used in each step. Model were built as a fit-for-purpose model for PBBM applications, so it's involved a mechanistic absorption model and a 2-compartment disposition that were modeled based on IV dose, microdose data collected from 6 health volunteers. Here is the graphs and the results for the compartmental analysis. Model also included pre-systemic metabolism data that were included as a percentage of first pass extraction, hepatic and also intestinal, with slightly different values for the fed and fasted state. Hepatic intestinal first pass extraction were arbitrarily attributed as being half of the hepatic value. So we questioned the company about more data and the knowledge about the metabolism of the drug. So it was presented that the urinary clearance is negligible, so the total clearance can be considered as equal to the hepatic clearance. Results from a mass balance study that were available after the modeling also corroborates the values of the FG and F8. Results from a DDI study with itraconazole also reinforces that there is a limited contribution of CYP3A4 for the hepatic and intestinal metabolism. And regarding transporters, also results of a DDI study supports that it has a low contribution and is expected to be saturated at the dose level of 500 mg. But we consider that a parameter sensitivity analysis on the first pass extraction percentage would be informative for the modeling. Here I present the table of the pharmaceutical pharmaceutical properties included in GastroPlus, highlighting that they used the default physiology fed and fast stated. And here the table with the compound tab input parameters. Highlighted in yellow are some key parameters that we are further discussing. So log D is included as between 2 and 3. pKa were modeled as 4. So compound A is a hydrochloride hydrate salt, so it exhibits a decreased solubility in the presence of chloride ion. Taking the average value of the chloride concentration in jejunum, company consider more biorelevant to include in the solubility median a fixed amount of sodium chloride as 100 millimolar to extract the solubility data. And this is the values that are presented in table 1, which shows a very low solubility data for this drug. Highlighted in yellow are values obtained without sodium chloride. And in media without sodium chloride, this drug exhibits a profile of pH-dependent solubility, with being slightly soluble at pH 40.5 and practically insoluble at pH 1.2 and 7.4. But we question if this approach for modeling the common ion effect is adequately justified. Company claimed that these were a worst case scenario as they decreased the solubility of the drug. Based on this value of solubility with sodium chloride, they fitted a pKa value. And the pKa resulted in a value approximately 4, which is quite different from the value obtained from ADMET predictor and also data that was submitted in another case. So there is a considerable uncertainty on these pKa values, which drives all the properties of the drug. And here's an important point: if experimental value would be necessary, and if parameter sensitivity analysis would be somehow supportive of this included value of 4. Regarding permeability, they had results of a CUCL2 experiment, and they used the GastroPlus built-in conversion tool to calculate path. It was estimated as 1.74. The model was calibrated with standards used in the experiment. They also performed a parameter sensitivity analysis, which demonstrate better results with lower values of PAF as 1 and 0.5. So we questioned if the estimated PAF for this BCS4 drug is accurate or if lower values could better explain the PK results. Regarding precipitation, there is no experimental data, but they used the default value precipitation time in the software, which predicts precipitation for the solution with a rapid redissolution. And for the tablets, there is no prediction of precipitation. This considering as input pKa value of 4. So they also present the parameter sensitivity analysis varying the precipitation times 10 times lower and faster, and there was no difference in the precipitation results. But as from the regulatory point of view, we think experimental results on precipitation would be informative. They included in the model lysozyme trapping after seeing that for modeling the late Tmax that was seen for every formulation, including the oral solution with 6 hours of Tmax. After considering that the dissolution, precipitation, and low passive permeability are not the root causes, lysosomal trapping would be a reasonable explanation for the late Tmax. But this would make sense if the pKa were considered to be around 9, not 4. So we questioned which other factors could at least partially explain this late Tmax, and if a parameter sensitivity analysis would be necessary for justify the percentage of unbound fraction in the enter site. Going for the dissolution, for the oral solution, they used the Johnson model, and for the solid formulation, they associated this with the API PSD data obtained by laser diffraction. There were no input of in vitro dissolution of the tablet, which we consider is not an acceptable approach as the particle size distribution is an API CKA that can be modulated or modified by process parameters and material attributes, so it would be necessary to include formulation CKA as the in vitro dissolution. Moving to model validation, model work validation using 5 data sets that encompass first-in-human studies, food effect, absolute and relative bioavailability, and data set 4 and 5 are considered the most relevant as they included TF2 and TF3 formulations. All simulations in the validations were done as single individuals. There were no variability, no population simulations, and no VBE studies. Simulation length matched with the observed data, the clinical trial. This drug has a long half-life, so some studies were truncated and others were not. And prandial state matched the clinical one using default physiologies. We consider that population simulations and VBE comparisons against the reference PK data set, which is TF2 formulation, would be necessary to support a regulatory decision-making. And also, a non-BE study or non-BE arm would be highly desirable. Here are some of the validation results. They use as acceptance criteria 3 ranges. If the ratio between the predicted and observed values were within 25%, they marked as dark green. If they were within twofold, it was marked as light green, and the above twofold was presented in yellow. So on table 11, we have the results of data set 4, and on table 12, results of data set 5. And indicated with the arrows are results of the market formulations, which you can see that is within twofold. Company concluded that the validations were adequate, but we questioned the acceptance criteria that were used, and the lack of graphical results for presenting validation, and also the fact that some results, especially in the single ascending dosing data set, were with above 2 folds with a great error. So this could have motivated some more refinements in the model. But moving for the model application were based on the D10, D50, and D90 values of the reference data set, TF2, keeping the constant ratio between these values. They performed simulation if the values were 0.1 and the 2 times above these values. So the fact of considering a constant ratio between the 10, the 50, and the 9 is a limitation of this model because other bets, other formulations have different distributions of particle size, as is shown in the blue table. They saw the results on application simulating different doses, 100 milligrams, 500 until 1000, but the dose of interest is 500 in the fed state, and for application, they consider 540 hours. So the results of interest is the orange line. Based on this, they simulated the value that would be around between the bioequivalence, considering the 500 mg results, and then have a mathematical value for the D50 odds of failures. And combining these results and keeping the ratio between the 10, the 50, and the 9 constants, they have the proposed specifications. But as I said, a limitation is that the results are directly applicable only for the TF2 PK dataset, and we have concerns if this would be applicable for the market formulation. So summary and conclusions. We think there is relevant uncertainties on key input parameters, mainly pKa, solubility, and lysosomal trapping. We have a major concern on the model development strategy that involves a direct incorporation of API PSD instead of formulation dissolution. The lack of variability on results due to the use of single formulation is another limitation, and the fact of results are directly applicable only to TF2 PK results and distribution. Then we think the model as presented can be only supportive and cannot justify any specification. As possible approach, we would suggest to obtain experimental results for pKa solubility in the physiological pH range without sodium chloride and with varied amounts of sodium chloride to build a link as a relationship or a correlation between API PSD and drug product in vitro dissolution, appropriately include in vitro dissolution in the model, ideally validate the model with non-BE results, preferentially due to variation of API PSD, include population simulations and adequate representation of within and between variability, and define the safe space for API PSD. So these were our recommendations for the company. And with that, I thank you for the attention and be available for the questions, and leave my acknowledgement for all the visiting that work in the case evaluations and also the academic professors that work with us.
Time for some questions, but please use the microphone, which I'm not doing, but please do.
Thanks. Hi, Amitav Mitra Kura. So question about the non-BE batch. That is, I guess, a thorn in my existence. So if I remember correctly, there were formulations with micronized, non-micronized batches. There was food effect study. There was a PPI study, if I'm not mistaken. So you have all these variations, multiple types of formulations, conditions. You still don't think, you still want a non-BE batch for model verification?
I pointed this as preferentially. I think if the company included the in vitro dissolution results and are able to validate the model adequately with adequate acceptance criteria. Maybe this can not be necessary, but we are not at this point with the data that were presented. So would be preferential, but if they could build a link or a correlation between API PSG and in vitro dissolution with the available data, perhaps this could be skipped.
Okay. All right, thank you.
So I think in a lot of ways, the dissolution of intermediate between critical biofarm attributes and in vivo performance. So if a company were to build a multivariate model between those PPAs and the dissolution, I think that theoretically we could kind of skip that intermediate step of using the dissolution as an input. So I guess like when you reviewed this case, I guess like was it just that the quality of that linkage was not strong, or would you say that having the dissolution is always going to be a requirement?
Yes, this case was a mock case, so we didn't have access to all the data, including the manufacturing process of the TF2, TF3 formulation. So there is no strong evidence that API PSG is the main driver of absorption and PK. And we believe that company had the dissolution data, and we think that the dissolution data, according to other data, is discriminative against API PSG. So we couldn't find a justification for not including dissolution and not having this further detailed in the reports. So would be the preferential approach, as this represents the formulation with the CPN's manufacturing process.
Yeah, this is Sandra Suarez. I'd like to make a brief comment. I don't want to promote session M, but I invite you to come, because I think that in that session, I would like to hear your input about whether we need only dissolution data. And so the moderators and speakers there will present a view, and we want to hear from you. And hopefully that people who participate there, we can, I wouldn't say reach an agreement, but have a good understanding of, the relevance or not of the solution to build the safe space at the end, because after all, is the ultimate goal, right? So, thank you.
So if I saw correctly during the model development phase, sorry, Joe Kushner from Pfizer, when they were doing the model building, they only used IV and oral solution data to build that model. So in some sense, there wasn't any API dissolution process included with those formulations, although I thought I saw with the solution, there was some precipitation and redissolution of API particles. Would there have been any benefit for the company to have provided tablet formulations as part of their model build to now incorporate the API dissolution process more formally as part of the model build, rather than just using those data sets for validation?
It's an interesting point. We didn't raise this, but would be beneficial since they have 5 data sets. They could have one for the model building and using the others for the validation.
A quick one from me. I found it fascinating when people don't have fitting the Tmax, they just directly move to fitting the FU. and you explain also, touch briefly on the low PEF. Was it then further looked at if low PEF was the cause, or some intestinal binding data was available to inform FU? And
the PEF that they used was 1.74. But in the sensitivity analysis, if they have used 1 or 0.5, they would partially explain the late Tmax. And we argued if these PEF values would be better but they stick with the path that were estimated using GastroPlus because thought that this would be a fitting. But we have to go further. We didn't have the access as well to the CACO2 experiment to see how certain is the experiment. But perhaps if they have a lower path, they would not be needing to include lysosomal trapping.
Because it's a BCS4, it's probably could be the case that the lower path is the case.
And also the rationale for lysosomal trapping would be feasible if they have included the PKA as 9. So we think that the main issue is the uncertainty around the PKA that would change all the drug properties.
Yeah. Thanks.
Last question for you, Rob.
Is that on? Good. This is Rob from FV. I continue to have myths about one can actually predict the solution without knowing the formulation process aspect. And even though you use a pure API physical mixtures, during the manufacturing, there may be over lubrications. Maybe there is agglomerations. Not to mention during the granulation, weight granulation, lower compaction, full bed granulations, this could have a profound impact. So I would like to know why maybe the modeling is confident about you can just use the API property without considering excipient effect, manufacturing effect. It could be the same formulation. If you change your manufacturing processes, the dissolution could be totally different. So I would just caution the industry to try to use a model and without considering this formulation process and thinking that we can actually predict the resolution accurately. I think there's a dangerous zone to be in. Unless the model will consider some of this formulation factor, manufacturing factor in the model. Without that, I'm just doubtful personally.
Thank you for the comment. I totally agree. I think this is a strong limitation of this strategy, and we miss it having this discussion on the solution and manufacturing process.
Thank you again. It's now my pleasure to introduce our last speaker for the case studies this morning. And Shinichi Kojima is Associate Senior Scientist for Clinical Pharmacology and Pharmacokinetics at the Office of New Drug 5 and Office of New Drug 4 at the Pharmaceuticals and Medicine Device Agency, PMDA in Japan. And he has certainly traveled very far away here. He is currently, he graduated from Meiji Pharmaceutical University since 2009. And since then, he has reviewed pharmacokinetics, clinical pharmacology, and pharmacometrics of new drugs. He also had a stint with the FDA 2016 to 2017 with the, as an All Rise Fellow in Division of Pharmacometrics, OCP, OTS, CEDAR, and FDA. And at the PMDA, he leads the modeling simulation project team, and he has significantly contributed to the guideline and on PBPK, exposure response guidelines, population approach guidelines, which are all published by the Ministry of Health, Labor and Welfare in Japan. And as some of you know, there is a Japanese PBPK guideline, which he, as a leader, was developing. So welcome, Kenichi, and please take the lead.
Thank you for your kind introduction. I'd like to thank you for the opportunity today. Today, I'd like to talk about the case study 6, and it's focused on the model validation and application. It's disclaimer. The content of this presentation is just my personal view. This is an outline of my presentation. The first thing that I would like to mention is that the information is a presentation is limited in order to protect of the company's intellectual properties. It's a request from the company. So detailed information and the product and the model and the model building process and on setting parameters and validation results are not shown. So, however, without showing that information, I believe that I can provide some information to discuss in the this workshop. So first, I'm going to talk about high-level summarized information of the case study 6, drug X. And then I'll talk about the overview of the PBBM models, about the model development, model validation, and application. After that, regulatory concerns from the case study 6 from validation and application point of view. Finally, I'm going to talk about the conclusion for question from the company. This slide shows high-level summary of the information of drug case study 6, drug X. First, the model objective of the PBBM presented by the company was, does the agency agree that X% of polymorphic impurity is allowed in the drug product in light of clinical relevant specifications? As a background information of the drug X is a BCS class 2 compounds and neutral species and physiological pH range and immediate release or dosage form. Description was shown from the company in this case study, like as context of use. It is a model was developed to justify the presence of the X% of polymorphic impurity in the drug product will not have any impact on the systemic exposure and clinical performance of the drug. Regarding model risk, no information on the model influence can be taken, unfortunately, but the decision consequence seems high, I think. This slide shows a very high level of the PBBM workflow. It developed a PBBM using compound and formulation specific input parameters, and then validate PBBM by comparing predict versus object PK data for the different formulations, and then assess the clinical impact of polymorphic impurity as an application. I try to present the information to the extent permits from intellectual properties point of view as step by step. First is model development. As compound-specific parameters, molecular weight, log P, PEFT, solubility in media was considered, and distribution and clearance parameters were derived by the POP-PK model. As formulation-specific parameters, dissolution profile was integrated as a defactor derived from physiologically relevant dissolution testing, PBDT. And novel workflow developed to assess the impact of the polymorphic impurity in the PK is used, but I cannot share the detail of that in this presentation. Next is a model validation. Model was validated against the clinical and non-clinical variable for the as a critical quality attributes. Data for the validation was including clinical data from different formulations and different dose levels, food effect study, and non-BE relative bioavailability study data, and non-clinical data from dogs. PBBM model could predict the in vivo relevance of the change in the formulations and process parameters. Here are some of the validation results. These results are not all, but may be helpful in understanding about this model. For the table on the left-hand side, information of studies, formulations, and those information are masked. But however, multiple study and multiple formulations and multiple dose strengths results were used for the validation. The ratio of the predicted result to the observed value were within the 10% for the majority. It's 90% of predictions are within 1.1 fold of in vivo mean PK parameters, and 100% within the 1.25 fold, 100%. And the table on the right-hand side compares the observed and predicted results of the non-BE reactive BA clinical studies. The results of also predicted non-BE result. Thus, so the predictive performance of the model for the both Cmax and AUC is consistent across all formulations and dose strengths. Next, I'd like to talk about the non-clinical data from dogs. Please note that we are unable to give you an actual positioning of this data as we are unable to the provider, unable to the provider all information. I think this situation is simplified in this presentation, I think. First, this slide shows some PBDT profiles for the polymorph 1 and polymorph 2. One thing to keep in mind here is that dog model was just to validate the specific assumptions, since the companies only had the data on the polymorph 1 and the polymorph 2 in dog, not sponsors have no data in human. The PK parameters used in the model were specific to the species model. They did not use the dog model to estimate any parameters for human model. From the figures, as you can see, the dissolution from the polymorph 2 is so later indicated as a blue curve compared to the polymorph 1 in red. And the PBDT indicated that drug X remains in a supersaturated state for at least several hours. This slide shows validation results for the comparison prediction results from the dog model to the dog observed data. I apologize for a mistake on this slide. The figure on the left-hand side is a result of the polymorph 2 instead of 1, and right-hand side is a result of polymorph 1. I'm sorry. So, yeah. the validation results were good, looks good, and the company concluded that the assumption they tried to confirm from the doc non-clinical data was correct. Next, this slide shows model applications of the PBBM. Validated PBBM model was applied to assess impact of the polymorphic impurity in the drug product on the in vivo exposure. PK was predicted for different percent of the polymorphic impurity, value percent between the 0% and 100%. In addition, Bachelor B study were conducted. The condition of Bachelor BE are 10 trials, crossover design, number of subjects is 20, reference product is 0% of polymorphic impurity, and test product is X%. This slide shows concentration time profile predictions for the different percent of polymorphic impurity. The bottom red curve is 100% polymorphic impurity, and the top blue curve is 0%. As you can see, the higher the amount of the polymorphic impurity, the lower exposure. So we can understand the effect of the percent of polymorphic impurity of the PK profile. This slide is a result of the Bachelor B study. A, B, C, and D assumed different percent of polymorphic impurity. The upper and lower limit of the 90% confidential interval for Cmax and AUC are shown respectively. And the result of the 10 studies are arranged vertically. Green cell meets the BE criteria, and red cell is failed. As you can see, that about D, situation D failed all 10 trials about the Cmax. On the other hand, the A and B and C were all green. However, it's just my personal opinions. Although C, pretty look like C and C looks C max. Although C meets the criteria of BE, many of them after near the lower limit, and considering the uncertainty of the models such as variability of the every parameters, it may be difficult to make a decision in actual reviews if this model is a high risk. If this model is a low risk, it's no problem. Yeah. B looks better than C, and A looks pretty good about the B criteria. This slide shows regulatory concern from our discussion focused on the model validation and applications. The first point of the role of the animal PK data in PBBM. Use of the non-clinical data is needed to fit for purpose. We do not believe the model building with animal data is optimal and should use human data whenever possible. However, the data from the dog model is complementary to the human PBBM for confirming model assumptions and the contribution and the conditions in the dot model are considered under the conservative conditions compared to the human conditions. In that case, use of the animal data may be acceptable. Second point is acceptance criteria for the virtual B study. As already pointed out in the previous case studies, questions such as how to assess variability, how to address the variability uncertainty, and what if some virtual B studies were outside of the acceptable limit. For example, we tried 10 trials, 4 trials failed. How to decision about the BE. So I think that this point probably will be discussed breakout session in this maybe this afternoon. This is my last slide. Regarding the company's questions presented here, the PBBM model shows a generally good predictive performance, and the model is therefore acceptable for the purpose of presented. However, whether X% is acceptable depends on the evaluated for virtual B study. For example, more conservative value of the X may need to be chosen if the variability setting is assessed in the actual review as uncertain. In the case of the model is high risk. Thank you for your attention.
Okay. We're going to get started with our roundtable discussion. I'm going to introduce those that will be serving on our panel as well as our moderators. So for the panelists, when you hear your name, please make your way to the front. We have Rebecca Moody from the FDA, Louisa Burgess from Envisa, Mary Matatari from MHRA, Laura Tusanuno from Belgium FMHP, Srini Virenshimi from Health Canada, and Shinichi from PMDA, Paul So from FDA, and our moderators will be Tycho and Claire. Okay, I'll turn it over now to our moderators to begin the discussion.
Yes, is my mic on? Great. Thank you all for coming back on time, and we're going to have an exciting roundtable. We have pre-prepared some questions, and we will ask the regulators for their input, and then, time allowing, we will also solicit additional input from the audience here. If you do end up asking questions, please make sure that you use the microphone. And for the panel members, you have to push the button to make sure it's green if you want to use the microphone. Now, we just have the pre-prepared question. I'm going to start it out, and then Claire has the next questions. First question is, if we can advance the slide, what can sponsors do to overcome non-availability of intravenous data to validate PBBM model? E.g. independent clinical study arm or solution data, etc. So what do you do if the sponsoring company wants to do high quality drug product quality involvement with PBBM, but they don't have IV data? I am pointing to Becky, to you first.
Told you your question was going to get answered today. Yeah, I know that IV data is not always something that can be done during a drug development. It's not feasible for all drug products, and considering generics as well. If you don't have IV data, we like to see oral solution data to help build the disposition model. And then you can use other studies, DDI studies, mass balance studies, single ascending dose, multiple ascending dose to support any parameters or justification, selection. So yes, we encourage IV, but we understand that it's not always going to be the case.
Wonderful. And to you, Flora.
Yes, given that these are pre-prepared questions, I also have pre-prepared answer. Yes, my answer to the easy one is actually that I don't have an answer to this question. But I'm not sure also that what would be most helpful here is to come with a sort of cooking recipe, like that would work with everything. I think that given that we were saying yesterday that we in the learning curve, we think really to prioritize interaction with regulators at this point in time. And in particular, when comes our recommendation and experience from EMA perspective in particular, we really prioritize this credibility approach, which is the reason why it is on the screen. It is important before we look which data do we have for building, which data do we have for validation, is it enough, is one patient enough, or do we need more? We need to locate the modeling in the totality of data that we do have to support the answer. So the risk-based analysis is important. And in the risk-based analysis, for us, we have a term that we call regulatory impact. It is actually the place where the role the model plays into the answer to that question. So it is important to know the context of use, the actual question, the model influence, the decision consequence, but all of that will end up with something that we call the regulatory impact. When we know whether it is important or not, we have had case studies yesterday and today, where for some time it was important, for some time no. Based on that, then we would go in that checklist that Mary was referring to in our presentation. And then we are in the lower part of that actually slide, where we would look at the acceptance criteria. This is something else that we need to discuss with regulators. This needs to come from the industry, being agreed with the regulators before the data are actually analyzed. We need to make sure that given the question, given the context of use, would we agree having non-BE, not having non-BE data? Once we have agreed on that, then we go to model development, validation, every complicated stuff that you were talking about today. To me, they come at the very late stage. Sometimes we can stop at the level of low risk, no need for model. Let's save our time, both sides, I mean. Thank you.
Thank you, Flora, for that comment. Since we have the credibility assessment up here, that one was originally from the FDA and mainly from Office of Clinical Pharmacology authors, and I liked it a lot, but it had in it model use and model criteria, I believe, and there were DDI specific case studies where it was basically pointed out the basic concept was the more risky the decision is, if it's wrong, the more data you needed to provide. And I wonder if you're going to get traction with regulatory agencies, then we should modify that manuscript, maybe a version 2, where we're then not talking about DDI applications and pediatric applications, but maybe have more like a formulation specific topics where we could just custom tailor it for that application. I think because when you read the paper as it is from Colleen Kummer, it was very well written. But if you give it to people who are doing the solution data modeling, it might not quite resonate. So that's my proposal. But thank you for this. I know I took a lot of time, so now it's your turn.
Okay, all right, thank you. So the second question is, and actually this question goes to Paul, first of all, how do the agencies consider model influence and decision consequence for setting model validation criteria? In effect, what is a model being used for? Should we consider it the same for all the models? So if you'd like to comment first, Paul, your thoughts.
That's kind of a loaded question, but I'll try.
Yeah.
To kind of go back to what Tycho was saying in that manuscript on the credibility framework, we take lead from that when we build models, not just in the context of PBBM, but PBPK and really all models. There's a lot of good information there. It's a framework, so it's really just setting up kind of guidelines on how to consider and approach it. I think originally it was for medical devices, and it's an ASTM or ASME standard. ASME standard. Thank you. So there's a lot of good information there on how you approach, your model, and it begins with, kind of the implication of what that model is used for, the context of use, question of interest, what have you. And then that helps you build out the risk and the risks to the patient. What happens if this thing is wrong? We really take lead, not just for verification and validation standpoint, but from, what your drug is supposed to do. So what does the clinician have to say? If the model is catastrophically wrong, meaning, it's not predictive at all in our case, what does that mean? Are you putting product out there that's sub-efficacious, or is it increased AEs, what you're looking at? So in that case, the risk paradigm changes. So that's really the way we approach, verification and validation of a model, not just for PBBM, but for really all in silico models.
All right, thank you, Paul. And perhaps we can ask the same question to you, Louisa. What are your thoughts for Anvisa?
Yeah, I think complementing this first answer is important to assess what the model is designed for and what would be the usual data that would be needed for that application. We have seen cases of biopharmaceutical risk assessment for some post-approval changes that normally would be answered by the solution data. So if the company proposed a PBBM, we could see this as a stronger data set than a single dissolution in theory. But for risk for when you are trying to bio-waiver a situation where a BE study is required, a PBBM can be seen as lower evidence with more uncertainty than the real BE study. And this can be taught as in the decision consequence and maybe inform the data set that is needed for the validation. I don't know if the mathematical criteria for considering the model validated, but the robustness of the data set can be informed by this risk of the application.
Okay, thank you. And perhaps, Junichi, you'd also like to comment from your side.
Yeah. So in the PMDA, the PBBM applications is handled by modeling and simulation project team, basically. And in the assessment process, before the assessing the validation result, before assessing validation result, we discussed with the CMC reviewers and review team about the COU of the modular PBBM applications. This is a very starting point about the for the assessing the PBBM application, because as Flora mentions, model how to evaluate the MITT application is based on the COU and model risk. So first of all, we discussed about the COU and model influence and model decision consequences, with especially the about the PBBM applications. We pharmacometry and clinical pharmacology is discussed together with the CMC regulator in PMDA. Thanks.
Okay, thank you. No, I think you're absolutely right. There's a lot of different cross disciplines required in the field. And then, well, last but not least, Mary, perhaps you'd like to give your thoughts on this question. Thank you.
Okay. Okay. Thank you.
We can hear you.
That no, I agree with what Paul said, what Louisa said, and that the model risk that is a function of model influence and decision consequences, as not only in PBBM but also in PBBK, even in population pharmacokinetics, the model risk is the one that drives the selection of credibility activities such as verification and validation of the model.
No, that's very clear from you all. Thank you very much for setting us in the right direction. All right, Tycho, question 3 for you.
Yes. Here we have question 3. Is there a minimum number of data sets recommended for model validation? Consideration for context of use, application needing to qualify the model validation, etc. I know we have listed here. Shirini, would you like to comment on question number 3?
Sure. I think I can start by saying that there are no widely accepted or agreed on recommendations for a minimum number of data sets required. But one of our aims during this workshop is to discuss these issues. And there's quite a bit of discussion between industry and us when it comes to things like this. So I think it does require some discussion. So going back to the responses that we just heard for question 2, one of the first things we do consider is the model risk, the regulatory impact, what's the decision that we're looking to make, the degree of uncertainty around that decision. Are there other data sets within the submission that support this kind of decision? What would we normally expect to see? If we didn't have a model, what would we depend on? So these kind of considerations are what we look to try and assess what kind of data set do we need and what kind of confidence do we need in this model for the intended use. I could point people out to the breakout session on model validation later this afternoon. I'm sure there'll be robust discussion on this. Thank you.
I think Kim also had a comment on this.
I think Shirini summed it up pretty well. I guess the only thing I would add is it's evaluating the confidence of the model. So the method validation needs to support that. So I don't think it's going to be a one size fit all. I mean, I know we don't have a concrete answer, but we have to evaluate it, based on the use case and the confidence that we need in the model.
Thank you. Back to you.
Okie dokie. All right. Question number 4. And I'm wondering, because this is also probably a pretty loaded one, perhaps we should split it into 2 parts. Or perhaps people would feel comfortable commenting on both bits together. So I'll read the whole question and then we see how we go with the answers. How's that? So what are the agency's thoughts on how essential is a non-BE batch for model validation? Or is it case by case? I suppose it goes back to what we were discussing before. And I think it also came up in some of the case studies this morning. I think, Tycho, you also raised the point in your case study. Also, how far do we have to go outside, or how far do we have to go to make a non-BE batch? Do we have to go extremely outside our GMP space, if you will, or make a wacky batch, as we often call it in the industry, make a wacky batch that actually has, so far outside it would never actually be produced, but that's where we have to go to be able to get that non-BE batch. And how relevant would that batch be? So we'd love to hear your thoughts on this. So perhaps Mary, you'd like to go first on this one.
Okay, thank you. Non-BE batch, known also as bad batches, are preferred to demonstrate model robustness and propose a safe space boundary beyond that is supported by bioavailability or bioequivalence data and within the model validation range. Now, the non-BE batch should have meaningful changes when compared to the applied finished product, and these changes can be at formulation level, for example, quantitative proportion of excipients may change, or the CMAs, for example, the particle size of the drug substance, as we saw today, can be changed or slightly modified critical process parameters. However, it is acknowledged that validation of the PBBM against non-B variants sometimes may not always be practical. This is acknowledged. Thank you.
Perhaps the same question to you, Shirini.
Sure, I totally agree with what Mary just said. One thing that we look for in non-bioequivalent batch would be if a company is really seeking to set an edge of dissolution failure. In that case, it's kind of helpful to have a non-bioequivalent batch and be able to not extrapolate beyond that. But in our case, we generally do not look for a non-bioequivalent batch for every single PBBM that comes in. In a lot of cases, it's quite okay to validate within the bioequivalent range if it suits the intended purpose.
Yeah, so I think to both of your points, it actually goes back again to the intended purpose, the intended question of the model. Yeah, but it's very interesting to hear your thoughts on that question. Thank you. Okay, Tycho, back to you.
You're very efficient, so we're going to have time also for the audience. So you can hold your questions, but there will be time. Question number 5, we have a few more participation and questions here. In particularly, of course, I represent industry, and we are wondering what can regulators do more of to promote and encourage innovation, particularly around PBBM use and model-informed drug development in a global environment. The reason we ask the question is because we often get the question, company internal, that it's nice if we submit to one agency and we get the PBBM model accepted, but then, somebody else may have a different opinion, and then we have to run the clinical study anyway. So then this sort of defeats the purpose, and then you have company internal low impact. Okay, so the question is, what could be done to promote science and also drug product quality to have more PBBM models submitted? I'm just going to start with Luisa. How about you?
Thank you. Yeah, I think this question is essential, and the agents have to focus on the patients and have sure that they have the right questions proposed that the model could explain as a tool or as other tools. So if we set that we need clinically relevant specifications, model can be able to apply to answer this question. And I think collaborations like this one is very important for us to harmonize the knowledge, and they having discussions with the agency as soon as the company has the intention to present the model. And also speaking from the perspective of an agency like Anvisa that has less structure in pharmacometric fields, is very important to have collaborations with academia or to put together PBBM with other MIDD discussions for having a strong knowledge and harmonizations in the field. So I believe that the focus should be the patient, not strictly guiding the science around PBBM modeling, but the agents be up to date with the scientific discussions and be open to discuss these strategies with the company would be a good way forward.
Thank you. And Shinichi, if I can ask you also to comment on the same question, what can be done at PMDA to?
Yeah, approach PMDA in order to promote the use of the PBBM for the regular decision making. It is necessary that the idea be widely understood by review team in PMDA, not just PBBM expert or the modelers. So for that, it requires further experience in the actual review process, because the case study for this workshop were very good experience, but the situation was different from the actual review, I think, because the because I mean in real regulatory decision making, not only the result of the model application, not only the result of the model application, but also the relationship with the results of the other all existing information. So it is very important. So I think that a little bit track of the case study, limited this type of the information. So I think the different, a little bit different, but between the case study and this case study and actual case review. So we need more experience in the actual review of the PMDA. Additionally, discussion with the regional Japanese stakeholders also is required, I think. For unknown reason, I don't know, for unknown reason, submission of the PBBM application to the PMDA, it seems to be lower than the other regions, especially the United States. I don't know why. So I think that so in order to promote the PBBM use in Japan, it is necessary to address its issues jointly with Japanese stakeholders. Next is international harmonization. I believe that this workshop is related to activity, are very good starting point for the harmonizations. I believe that a continued exchange and discussion of the information and opinion is needed within the global regulators. Thanks.
Thank you, very helpful. I know you have the PBBK guidance, and maybe down the road there might be a PBBM one, and you have more submissions. I'm going to go over to Paul. Same question for you. What can you do?
How much can I give you the long answer, or do you want the short answer?
Oh, I think we have some time.
Okay. So I gave it a lot of thought, and first off, if I could hold up the disclaimer, this is my opinion. But what I would say is, what the regulators can do, there's not a whole lot what we can do, I would say, because our hands are tied a lot. But some things that we can do is probably promote more guidances from each of our respective agencies. I think not because we are experts and we know a lot. Maybe these guys are, but I know nothing. Mostly because guidances in a way serve as endorsements of what we're willing to see and accept. So I think that's probably helpful to you all. The other piece, I think, and this is a long-winded part, is what I've learned over the past few days and over my career is biopharmaceutics really is the Rosetta Stone, so to speak, in the drug development world. We I don't want to say bridge, but we're able to speak the language to CMC, to Clin Pharm, to the clinicians, and bridge that gap and help folks understand from a drug development perspective what we're trying to do. So that being said, I think knowing your audience and framing and providing context of what you're asking for is helpful. And having an example, I have 2 young children. Not that they're smart, but what they do is whenever they want something, they frame it in a way that they're going to get what they want. So, for example, my daughter, she'll say, "Can I get this teddy bear because it'll help me sleep in bed better?" I got you. Or my son, "Let me eat this bag of candy because it'll help me do my homework." And those are, I'm not saying that's a smart thing to do or I endorse that, but knowing your audience, I mean, children know it. So I think and again, talking to many of you over the past few days, I think we all struggle with a lot of the same things, even on our side of the bench here, where you have many disciplines that you're trying to speak with. For example, clinicians, they don't always see eye to eye with us in terms of what we're trying to do with a modeling perspective. So I think from an agency, perspective, what we can do to kind of help things along is instead of just, for example, in an IR or when we request information, to say, we need to see more PK studies. Giving you a little bit of context. I know most of you in the audience probably know why we want to see that to support the model. But if we frame it in a way that it'll help you with the rest of your organization, I think that's something we can do, just provide a little context. We need to see more PK to be comfortable with the model to ensure we're minimizing batches released. We're minimizing AEs for batches released. You know, that might go a long way. So that's something we can definitely work on. And again, this is all my opinion, so please don't run off with it. But I hope that helps.
So know your audience and basically package it properly. But I do like the concept of when we get replies back that we have to think about our audiences too, right? So we have, like you mentioned, CMC, we have Clean Farm, we have formulation departments, and there might maybe other players involved. So if there is some feedback that says, okay, can you provide more PK data or can you include more PK data? That actually helps us because this is something specifically helpful rather than just saying the model is not appropriate or something like that. And I'm just going down the list. I think we have more feedback. How about you, Becky? Do you like to comment?
Yeah, I echo everything that everyone has said already. I will say, you know, our experience is only as good as what we receive. So in order to encourage more submissions, we need more submissions to fine tune what we want and what's acceptable. I think these case studies, getting together with the other regulators, sharing our knowledge was invaluable. And I would wholeheartedly appreciate more efforts to do that. And I think, you know, from our perspective in BioPharm, we are doing a lot to educate ourselves with the different modeling platforms and making sure we're staying up to speed and trying to give everyone experience with how to review a PBBM submission. And that's something that we're doing. But all I can tell you is that we want to see the model, even if, you know, there's so many models that are being built and being used internally, but are not submitted to the agency. And, you know, and I understand that it's because it probably is not good enough for a regulatory purpose, but for us to see the model and get more experience on how that was helping you decision make for formulation development could provide benefit down the line in your product development.
Wait, you want to see the water? That's fantastic. You know, that's really good.
It's my personal opinion.
Of course, everything is everybody's personal opinion. Flora, how about your personal opinion?
Oh, my personal opinion is on the slide. Can you put it back on? Yes. This is my opinion. And no, not that one. Oh, yeah, this one. This one? Yes. I think that what we can do from regulatory side, but the colleagues have already said that in a way or another. At least the way I see it is, first of all, train ourselves so that we can follow the story and give guidelines. Give guidelines to the industry so that they are also clear on what they can expect from us. But the reality is that actually the industry is the driving force. This is my personal opinion. This is why this framework is on the screen, to tell you that actually, because this PBBM is actually like the newcomer, we already have the old models for PK, PK/PD, exposure response, and then now you come with this PBBM obliging us to learn about precipitation. I'm fine. But the thing is that you need to learn from those that have succeeded before you. And because you are the driving force of this, you also need to communicate in such a way that you put the regulator in a comfortable position to support you. If the regulator do the step to learn, to stay here 3 days, hearing your solubility and precipitation story. Your side, what you can bring to all of this is help us not giving the impression that you are hiding something. Be transparent, communicate correctly. You come with a submission, start from the beginning. What is the impact? What is the actual data you have? Even your batch, the one that you were talking about, that is the one bed, how did you say? The wacky one. Put it on the table because you have it. Not only come with the fancy one, the one that look all perfect, so that you will know that regulatory like this kind of good plots. I mean, this is not the way to go. Contextualize it, come with argumentation, discuss the acceptance criteria before looking at the data. And then when we have all of that well set, we are discussing science. When we arrive to the data and the batch and the lot, we have agreed on everything. It's easier for everyone, learning and confirming at the same time. But if we have all the time half of the story, and then we're lost into 300 pages of stuff that no one understands, this is not helping us to help you. This is my opinion.
Yeah, so this makes, the clear strategy, the clear communication, so you don't get overwhelmed because you already have POP-PK on all these other models, and then you're supposed to be understanding PBBM. To me, it sounds like a little bit what also Paul and the others were saying is that we have to know the audience, we have to make it easy for you to digest, but also openly communicate about what I'm hearing here very clearly is demonstrate and communicate and say what the impact of the model is if it fails, perhaps. So very powerful. Now, and we do have time. We do have time, and anybody is welcome. There's lots of questions.
We're going to be running around this room in a minute. So.
I was strategically standing by the mic. So great panel, thank you. It's a great panel, thank you. I think the one thing that I took out of this is biopharma is the Rosetta Stone. So thanks, Paul. I'm going to use it. So if I want to probe a little bit about the IV PK a bit more. And I was glad that Heiko presented his case study. That case study had solution data, multiple doses. They had bottoms up, allometry-based clearance, rodent and non-rodent. And I believe he said human clearance were similar, very similar, something to that extent, and POP-PK-based estimates too. So in your opinion, in that case study, again, would your scientist and regulatory had both, would you require IV data?
Thank you for the question. Yeah, in the experience of Anvisa, besides the case studies that we are discussing here, we have a same case which uses a similar approach of using POP-PK and using allometric scaling. So it's case-by-case answer, but can be a reasonable approach in case there is no IV data or oral solution data. Can be acceptable, but depending on the case.
Yeah, fair enough. In that particular case, that's why I picked Tycho's case. In that particular case, you feel that's reasonable without IP.
I didn't have time to look at this case in details, but I can say that from other cases we assessed, it sounds a good approach.
Anyone disagrees with that?
I don't disagree, but I do have a couple things to add, if I may. And this is more outside of the scope of the case study, but just a general something I noticed. Not that we require, but recommend. I mean, IV data is kind of the not gold standard, but it's really highly recommended to use that to start, building the spine of your model. In the instances where you can't get it or it's not good or what have you, I've seen many creative ways where you can pull useful information, in particular, for example, these, I don't want to say non-B, but less equivalent batches. For example, again, and I apologize to our generics colleagues in the audience, but in an innovator application, you guys are putting in anywhere from 30 to 70 DDIs. That's a lot of in vivo data. If you're creative with it, and you do a little bit of in advanced planning, you can almost bake that in to get that information. And I guarantee you there's going to be a non-equivalent batch in there, or data that you can use to build your model. Is it optimal? No. I mean, it's better to have that IV data up front, but you know, there's a plethora of data that you can use. You just have to be creative and plan a little bit.
Since I have the soapbox, can I ask another question?
Go to Javier.
Thank you. A question regarding non-BE batch requirement. Would you think it is acceptable? So, Paul, you mentioned to look into to leverage all the clinical experience. And in that spirit, would you consider acceptable to use non-BE conditions of administration? For example, if you have a drug which is sensitive to pH, you dose it with and without PPI. And of course, the PPI, if there's a strong effect, you know, you may be non-bioequivalent to the fasted state without PPI. So if your model is able to predict this, would that constitute a case where you can predict non-bioequivalent, and this is related to in vivo dissolution in the stomach because you've raised your pH in the model? Would that be acceptable?
Can you just talk to me in the hallway? I'm just kidding. But I don't want to say it's acceptable, but there's a lot. That's a nuanced question. In general, I think the approach could be considered, but it'll depend on a couple things. I don't know if you're looking for a yes, no, but if that's helpful.
Yeah, thanks for feedback.
It's not a question, it's a comment, and I would like to add on what Flora was saying about sponsors telling the whole story. It's also about the timing, and the timing is not only at the time of the submission of the marketing authorization application. It's probably even more important, more critical, that this takes place early on on an early dialogue in the context of I don't know, scientific advice, as we call it in Europe, or the equivalent in other regions. That would also help both the regulators, but also the sponsors, applicants, to engage and to create that rapport and that understanding about what's going on and what are the purposes. That comes from a kind of regulator's perspective. Yeah, by the way, I'm Evangelos for the European Medicines Agency. That's all. Thank you.
Sumit Arora, Janssen. I think Evangelos just took the words out of my mouth. I was really going to touch on the fifth part. With all the clear strategy and clear communications we are talking about, are we going to see some faster channels from you guys where we can interact you faster? We don't have to do lengthy briefing books over 40 pages. We still can be transparent. We still can be agile, but how can we then communicate faster and get feedback? Because we still have to influence the program, and the clinical program sometimes runs really fast. So how can we interact with you guys better from all?
I would like to promise you that I will create a new procedure for you guys only to come and communicate. But unfortunately, this is really beyond my control. And you will need to use the channel that exists. And this is something very particular to modeler. They always like to have their own rules. No, I mean, you need to come. I mean, from EMA perspective, like Evangelos was saying, we have the scientific advice procedures that are really very modulable. You can come with one question, and then the format is you have your question, you give your own opinion, and then you tell us that this makes sense. Normally, when it makes a lot of sense, we end up saying yes. I mean, I don't think I can be easier than that, really. But in some cases, we also have other procedures like qualification of opinion, qualification advices, where you can come with a principle to be applicable to several drug development programs. Like you want to set up an acceptance criterion. I mean, this is something that we can discuss at scientific level, at methodology level, and agree on that. This exists already. Your companies come to that channel for validating biomarkers. You can come also using exactly the same channels to qualify some of the criteria you want to use in your PBBM. These are there and they can be used before marketing authorization application. And I can only speak for EMA, unfortunately. Mary?
Yeah, I think for MHRA, a scientific advice meeting would be the channel to communicate with us.
The question always comes how agile we can go, how faster we can go. It's always the time component there. So if you are waiting, let's say for 4 months, sometimes that's not fast enough. But as so just a thought that we need to be a little bit more creative how to move this field forward.
Can I just add that at Health Canada, since we are a pilot team that's dealing with PBBMs right now, we're quite agile as well. So you can request a pre-submission meeting, but the advice you get is often based on how much you're willing to share in a briefing package. So putting together a very short briefing package really would not be to your benefit, maybe.
That's clear. I think it's really useful. Do you hear me? Yeah, really useful to hear this. I think to your point, we also have to find the best way of creating that transparency to share with your good selves. At the same time, you also want to give us that direction in a good way. I think the question is a fair one. I think it could also be a topic for further working on. Where's Greg? Round 2 of this workshop, Greg. Could go. No, I think it's fair, and I think we should work on it together on how to make that communication process more agile. Yeah. Yes.
Yeah, this is Michael Wang from Merck. So we understand, use of human PK is ideal for model validation or parameter assessment. But we all understand it's expensive and a very lengthy process to get it done. So I'm glad to see Shinichi-san give an example of using animal data to validate the model. So I just want to know, other panelists' just opinion regarding use of well-controlled animal data as a tool to validate the model or doing some parameter assessment.
Yes, we have FDA seen cases where they use animal data for validation or, justification for some assumption that they've made. And favorite phrase, case by case basis. But yes, it depends again also on the context of use and the model risk and where the animal data is coming into play.
Joe Kushner from Pfizer. So I have a question related to the philosophy around the model workflow. So Mary, in your presentation, you showed a very simple high-level workflow, which starts off with the question that we want to ask, and then we go into model development, validation, and then the results. I think from a practical standpoint, at least in our development, we're building a model throughout development as we get additional data. So we're doing model development, model validation, not necessarily with a specific question in mind. And then we interrogate that validated model for specific questions. So if we want to do a design, a dissolution safe space, that's a question we're then asking after we've done model development and validation and all of the PK data sets. So I just wanted to ask the question of the panel from a philosophy standpoint and the way we do model development from that workflow. Is it necessary to be designing the model to answer a specific question, or can we have a general model that's built across development that we're then applying to answer specific questions for a regulatory purpose?
Well, like, first of all, the over the chart I show, it is the one included in the FDA draft guideline. I think maybe this flowchart can be used to when you do your submission to regulators, and then you have a specific question to address.
I guess in that case, if it's a specific question, we may not need all of the available data sets to answer that specific question for a model build.
Yes and no. So what I'd say to that, again, this is my own opinion. If you're doing the exploratory exercise internally, it's up to you if you want to have that question laid out ahead of time, and then you go and refine your model and do that process. But once, at least for FDA, I can't speak for everyone, but when we see it and you submit it, we want to see that question up front. So once you put that in there, it kind of, I don't want to say it locks you in, but it directs how everyone thinks. I mean, take that as you will. Your mileage may vary depending on a couple of things, but
I would like to contribute on that one. I think that the message here is that the scrutiny the regulator would look at the model you submit will depend on the question. It will depend on the context of use. Really, that's actually the point. So if you think that you have a minimum requirement internally for your company so that all your models need to pass that bar, if it's good practice, why not? If you always have a perfect model, even when you have no impact at all, this could work. But when you submit it to regulators, I mean, from EMA perspective also, the starting point is the impact. The starting point is the question, the context of use, what is the role of the model in the whole decision-making. Based on that, we would be looking at your model differently. So one model can be acceptable for a particular application and be refused, exactly the same, be refused for another one. So if you have that in mind and you decide to take like 2 months advance, already developing your internal model, I don't think that is best practice, actually. Thank you.
Yeah, I'd like to provide opinion, given that I have experience with the FDA and now part of a company, but I think I want to send that question back to the industry. From looking into the future, you want your model to answer only one question, or you want your model to answer any kind of questions? So if the model has been built a priori has been planned, then perhaps you will have a very strong model, robust model that is the data. Perhaps the data was used to build an IVIVC on an IVIVR, and as for regulatory agencies, if the model supports an IVIVC and IVIVR, then as for regulatory guidance, EMA, FDA guidance, it can support whatever changes, right? If you build it with 2 formulation variants, you can apply it for this. You can build, if you build it with 3 formulation variants and you did external validation, it's going to be a very strong model that is going to support major changes. But if you're telling the regulator, I built the model with an IVIVR and it is not fit for purpose, the validation, then perhaps the regulator, my opinion, right? The regulator would say, for the question you're asking here, which perhaps is a side change, perhaps the risk is low because it's an, even for a modified release formulation, perhaps the risk is low. Perhaps I could use, this is my own opinion, I could use the model for that specific question. So that's why, from my point of view, you need to specify very clearly upfront that the model is going to be used for this specific issue. But from the regulator's mind, if they see that your model is very robust, right? I think as part of the report, as part of the regulator report, I would say that this model is robust. It could, for future, right, for model maintenance, right, and applicability for post-approval changes, that then there is a good understanding that the regulators know that the model is robust and you could use it for any changes if they are supported by all the whole data, the whole risk assessment, right? Because you have heard one common thing from the regulators. They use also the totality of the data to make a decision on that specific question too.
I have.
Hi, this is Wilbert de Witte from SQ Labs. I was wondering, after it was mentioned that the regulators follow the science, and also after the keynote lecture where it was already mentioned that the ideal will be to also include PD in this whole exercise, what the regulators' opinion is on maybe some flexibility in the acceptance criteria if we can show with a well-validated PD and toxicity model, of course, that the Cmax maybe might not be so sensitive towards driving toxicity and PD in a certain application of a drug.
Okay. Okay. I will show tomorrow in the presentation with the case that was submitted to the Anvisa that we have received a case that was a combination of PBBM with PD. And although it was aimed to have a strong support for the clinical impact of the model, perhaps this is not the question that the regulator were made at the time. So the impact of the model also depending on the question that the agency is looking for. So in theory, if you have an integration of PBBM, a PK with PD, we are able to have more confidence in the model and answer several questions. But sometimes this is too much data for a simple question that could be answered by another way.
Yeah, I'm thinking more to accept maybe a certain formulation or a certain application that would not be accepted based on only the PK, but given the PD framework and the knowledge that we are, for example, working at a completely saturated occupancy with low toxicity, it might still be acceptable to consider that still as a good application, although the PK is maybe more than 10% or more than it.
Yeah, I think we're looking into PBBM, PD approaches as well, just for us to better understand the model risk. And the PD can help us understand the model risk if it's wrong or not. But yeah, we haven't looked into approving something where the PK is showing that great of a difference. But again, PD, I think right now, personally, it's supporting that if the model is wrong, there won't be a risk to the patient.
All right. Hi, this is Maitri from Cetera. I have a question like, what if regulator is recommending to perform the mechanistic model to justify a few of the application, and maybe it's a part of regulatory query. And then in this circumstance, how to approach the agency to justify or to communicate like regarding modeling approach and all, because it's always a part of a regulatory query, maybe it's a CRL or DRL. And then what's the way of communication to the agency to how should the approach for the model development and validation?
I got the question. I said, oh, I'm sorry. If the question you want to know if you have a sort of question which is beyond the data, how to approach the data.
No. So far we discussed like about the communicating with the agency well in advance, right?
Yes.
So what if like we get the recommendation from the agency itself, like you can perform the mechanistic modeling to waive this in vivo study, and when it's a part of regulatory query,
then what is the way to communicate to the agency? So let me give you one example. Like based on my prior experience, like I got one DRL or CRL from the FDA. And like in that, like if I decide to go with the modeling approach, then what's the communication channel to discuss the modeling strategy?
This is for FDA then?
No. This is general question. Like
actually for EMA, this is very, very easy. I mean, you can always ask for a clock stop. If you are in the process of marketing authorization application, and then you have question, and this has happened, you're right. That as part of the question that you receive from us, we say that this or that approach can better be approached by a modeling approach. If you want to discuss methodological approach aspects of what we're recommending, you can ask for a clock stop and come for a scientific advice. It is possible at EMA. And then we discuss for that particular question with the focus on the modeling and the recommendation. And when we solve that one, we have agreed on something, you can come back with that in the initial procedure. So this has happened, and yes, from EMA perspective, this is the way to go, at least. Thank you.
I would say, and to add to that, I would say stay in the swim lane in how you were responded to. So obviously, I mean, we're not going to open up a conversation out of the blue. So we're responding to something. So, I mean, if it was part of the IND, you stay within the IR channels of the IND. If you're in NDA phase or ANDA phase, you stay within that. I mean, it's very regimented in how we talk to each other. So I don't think we have to overthink that piece. I'm not sure if I answered your question.
No. Again, like, the time, again, I'll talk about the time and the speed, because when we receive the regulatory query, like it's a DRL or CRL, we have a limited time to respond. And in that particular limited period of time, like what is the way to communicate with the regulatory agency about the modeling strategy before submission or before responding that regulatory query?
Yeah, I don't know about a clock stop, although that does happen in certain circumstances. But I think most of the regulators, we're all human, so understand the need for flexibility. If you need more time, just say why, and you need more time. And most of the time, if it makes sense, could happen.
And do we have any channel, like after getting regulatory query, like how to, again, like I'm not like, I think I'm not communicating the question well, but what is the channel to communicate after getting the regulatory query? Because as per my previous experience, all the time I got the response from the agency that at this particular moment, we cannot discuss the modeling strategy or some technical part. And then if I am opting for the modeling at that particular point of time, and then how to connect with the agency and how to propose my modeling strategy before submission.
I can only speak for FDA. That specific answer will depend on the context of use that you set up. What are you intending to use it for? Is it just justification, supplemental information? Is it going to be pivotal? Are you going to use it in a waiver? Depending on what your use case is, then we're going to respond in the way you got, in terms of, we can't answer this now, it's too early, or sure, here's what we think. So it depends on how you frame what you're using that model for, at what stage you are in development and whatnot.
So the stage is after development, we have submission the product, and we have received the query from the agency, and that's the stage of the development.
What are you going to use a model for?
Maybe resolution, specification, justification.
Then maybe you'll get a response.
No, so like I have a couple of few experience like where I have not received a response. Like it's only like we cannot comment on a technical part at this particular moment. So and then we are helpless. Like how we can approach. Like we talked a lot about pre-communication and communication well in advance. Like before you start your modeling, you should communicate. But then what is the channel of communication when we submit the product, when development is done, and we receive the query after review? So.
Yeah, so it sounds like you have a very specific case that I'm not sure if I can provide you with an answer right here. Like I said, it depends on the kind of the paradigm and which avenue, if it's an NDA or an ANDA, and what the response is, what you're submitting it for. So I hear your plight. I wish I could give you clarity. But beyond kind of the generalizations that we've talked about, it's going to be hard to nail it down.
Thank you. I think the regulators have heard your question. It's probably really specific to you, so probably to give a general answer is going to be very difficult, but I appreciate your question.
Thank you.
Just in a principle of time, I'd just like to give the last question to Armin, if that's okay, and then we'll close the session, because it's been waiting.
Thanks, Claire. From very, very specific question to very, very general philosophy again. I think yesterday during the breakout session B, we talked about a lot about shift of paradigm, the way that we have to change our thinking, etc., which unfortunately didn't come across, in the report that was given. I think it was lost in the forest of the decision tree focus that we were having. But I think some of the discussion here is happening on the background that perhaps we are not appreciating as much as we should, that the case for model is no different than the drug. If you can bring a drug and, a year later, 5 years later, bring another indication for the drug, what is the difference with having a model and sometime later on say, I have got another application for the drug? The best way actually to look at this, our colleague in FDA, Leon, is not here, at least I haven't seen, but obviously he started to talk about this model master file like a drug master file. So I think we are talking about model as if any model, just we are starting from scratch and suddenly we are building this for this purpose. No, the idea is the whole of the drug development needs to be informed by a model, a general model, master model. And each of these master models, they will have new angles added to it. And the discussion for application, whether, the specification, etc., for that particular element, of course, it needs to happen in advance, that we are going to expand this model for this particular application by this approach, by, all the verifications that we have considered and get agreement pre-NDA, ANDA, etc. But we are not building these models suddenly from scratch for a specific thing. That is wrong way of thinking. The whole idea of, the incorporating modeling in all what we are doing is to integrate all what we know, all what people are talking about, the totality of evidence. Totality of evidence cannot be, sort of discrete pieces that they are sitting in different places. All of it needs to go into a model. And if a model is actually inconsistent in answering all those different elements, something is wrong with the model.
No, that's fair. Indeed. Thank you very much, Amin, for your thoughts. So we're a little bit late, sorry to the lunch cats, but I would first like to thank all of the panelists for bearing with us, answering all the questions, both the pre-prepared ones and also the ones from the audience. So thank you all very, very much.